My name's Dan, and I'm Head of Bioinformatics here at Greywolf Therapeutics. This talk is going to take you through some recent beta testing we've been doing with Spectronaut 20, particularly through an applied lens of how we've been using the software to increase the identification of these things that we refer to as ERAP1 inhibitor-dependent antigens. So to start with a little bit of background on Greywolf Therapeutics, Greywolf are a clinical stage biotech company. We are based in Oxford, and our scientific approach is really to target this signal one component of T cell activation. So T cell activation requiring a chain of three signals, where most current therapeutic approaches are focused on the latter stages in the signaling cascade, so both signals 2 and signals 3. But we really believe that by manipulating signal 1, which is antigen presentation, particularly in the context of disease, we're able to change the appearance of that cell to the immune system. And we do this by targeting two enzymes, both ERAP1 and ERAP2. And these ERAP enzymes are both immunopeptidases that sit in the endoplasmic reticulum of the cell, and their primary function is in peptide substrate cleavage prior to MHC1 presentation. So within any cell, you will have cellular proteins, and these cellular proteins will be degraded by the proteasome. And the products of that degradation will be peptide chains that will get transported into the endoplasmic reticulum. And then a proportion of those peptide chains will be cleaved by either ERAP1, ERAP2, or other aminopeptidases, and they will be subsequently loaded onto MHC. And it's this peptide MHC complex that then gets transported to the cell surface and presented to your T cells or your immune system. So that's what happens in the context of active ERAP. And obviously then inhibiting ERAP will prevent this peptide substrate cleavage. It will change what is bound to MHC and allows us to change the antigen repertoire on the cell surface. And we really apply this technology in two main therapeutic areas. And this is a little bit of an oversimplification, but our approach in oncology is to increase tumor cell visibility and tumor cell killing by turning ERAP off with our inhibitors. And this really is achieved by both an increase in T cell fitness, but also our ability to drive new neoantigen exposure at the cell surface. And then the opposite is true in our other therapeutic area, which is autoimmunity, where autoimmunity is driven by this misrecognition of self-autoantigens. We're able to apply the same approach and turn ERAP off and prevent the presentation of those autoantigens at the cell surface. And this really allows us an avenue to treat autoimmunity at the source of the disease. So it's perhaps then unsurprising that we make use of immunopeptidomics as a proximal biomarker for both ERAP1 and ERAP2 inhibition. And that's really because of where both of these aminopeptidases sit in the antigen processing pathway. So this peptide substrate cleavage is a post-translational modification of peptides, which means that we rely on observing these peptides in a mass spectrometer, particularly using an immunopeptidomics workflow. And what this immunopeptidomics workflow allows us to do, or helps us to do, is understand what the antigen repertoire on the cell surface looks like in an ERAP1 active state relative to an ERAP1 inhibited state. And that brings me on to the first of three case studies that we'll go through today, where each of the three case studies is used to evaluate the performance of Spectronaut 20 relative to Spectronaut 19. So the first case study is an early preclinical data set and actually the first project that we worked on with Biognosys to help us understand the dose-dependent effects of our inhibitors on the immunopeptidome of HCT116 cells. So to give a brief overview on the experimental design, this is a 6-series dose response experiment where we start from DMSO and increase the concentrations of the drug all the way up to 10 micromolar. There's an N of 2 per condition, and each of these samples is acquired in DIA-based immunopeptidomics. And additional to this, we take a combined pellet of all of these samples and generate a supplemental 10-fraction DDA spectral library. And then for completeness, all of these peptide searches in this case study and the case studies to follow were researched in the latest version of Spectronaut 19 and of course the new release of Spectronaut 20. And what this experimental design allows us to do is not only interrogate Spectronaut 19 versus 20 in the context of directDIA, but also in the context of hybrid libraries as well. So to start really with the results here on the left-hand side, what this left-hand graph is showing you is the number of peptide IDs at a 1% FDR from the respective searches. And a few things that became apparent quite early on was that when looking at these hybrid-based searches between Spectronaut 19 and Spectronaut 20, there was really only a modest performance increase or a modest increase in peptide IDs. However, when looking at these library free-based searches, we saw quite a significant change in this experiment. So the difference between this orange bar and this purple bar equates to about a 47% increase in peptide IDs. And what was also promising to see is that this purple bar in the directDIA search also now has about 25,000 peptides in it, which is not too far away from what we were achieving with the Spectronaut 19 supplemented with this 10 fraction DDA library. So this was really encouraging for us, and we went on to seek to understand if there were any differences or similarities between these two sets of peptides, really looking at this through a lens that it would be optimal to be able to design experiments in immunopeptidomics without having to generate a spectral library for every experiment you conduct. And the first thing we looked at is what you can see in the middle here, and is just how consistent are the MHC1 distributions from these two different searches. So with immunopeptidomics, what you would expect to see is this peak at 9 mers and this tail off at other peptide lengths. And this is exactly what we see here. So not only do we see consistency between what is Spectronaut 19 and Spectronaut 20, but we also see that they follow a nice consistent MHC1 distribution. And what that then transpires to is, because you've got an increase in peptide IDs in Spectronaut 20 and you've got consistent frequency distributions across all lengths, you get proportionally greater coverage in terms of absolute peptide IDs at each of those lengths. And that's again really interesting for us, but because we have some interest in those rarer peptide lengths from a biomarker perspective, particularly those out at sort of 12, 13 and 14 mers. So another thing we looked at in this data, and because this data is DIA and we're interested in making quantitative comparisons between the relative abundances of our peptides across different treatment conditions or different doses of our drug, we also look to see how quantitatively complete this data set is. So again, starting with Spectronaut 19 on the left here, we can see that we were getting about 15,000 peptides. And generally, most of those peptides, or generally all of the peptides in this data set, were quantitatively complete. So they had at least one quantitative value in each of our treatment conditions. And if you then apply the same analysis to the Spectronaut 20 search, again, what is encouraging to see is that this increase in IDs isn't just coming from spurious identifications. That number of consistently quantified IDs now increases from 15,000 to about 21,500. And you can take each of these respective sets and you can start to ask what the characteristics of the peptides are in each of the searches. So in an immunopeptidomics experiment, you would very commonly expect to see enrichment at these anchor residues at positions 2 and positions 9, which is where these peptides interact with the HLAs. And that is exactly what we see here. So not only do we see the enrichment of these anchor residues, but we also see enrichment of amino acids known to interact with the HLA types from this HCT116 cell line. And again, what is promising to see here is this, if you go and repeat this analysis for the Spectronaut 20 peptides, albeit more of them, the biology of these peptides seem to be relevant to the cell line we're studying. So a slightly different way to look at this now, albeit with a very similar plot on the left-hand side, is rather than ask what is the level of overlap between our treatment conditions or our concentrations of our drug, we can ask what is the overlap in overall peptides identified by each search. And what is interesting to see is that Spectronaut 20 and Spectronaut 19 here share approximately 15,000 peptides in their searches, and only 665 peptides are unique to Spectronaut 19. But actually, in the context of Spectronaut 20, what that is giving us is an extra 8,000 unique IDs for Spectronaut 20 searches. So you can again isolate that set of peptides, which are unique to Spectronaut 20, and you can ask whether or not there's any bias in these peptides or whether or not these peptides look like they're biologically relevant and usable for your analysis. And if you compute a similar motif plot to the one on the previous slide, you can again see recovery of these anchor residues at positions 2 and positions 9, and you can again see enrichment for these amino acids that we know interact with the HLA types of this cell line. So going slightly further now to try and understand whether or not we can recreate the biological observations we made in this data set from the effect of our inhibitors, what I'm showing on the left-hand side here is a principal component analysis plot from the Spectronaut 19 search, where PC1 separates out our biological replicates, and PC2 nicely and dose-dependently separates out variants based on the amount of drug we're exposing these cells to. And what is nice to see is if you again apply the same analysis to Spectronaut 20, you indeed get separation across PC1 that looks like it's related to biological replicates. And again, you get the same dose-dependent separation across the second principal component here. So starting with DMSO, 16 and 18 nanomolar, 400, 2010 micromolar. And you can also ask how many significant peptides there are in each of these different searches. So this is just computing a log-log model to ascertain what peptides will be significant at 5% FDR. So you take the log base 10 of your inhibitor concentration, you take the log to abundance of your peptide abundance, and this really just compresses everything into linear space, so you can start to fit linear models over dose. And what you can see is that generally both of the two sets of significant peptides are largely consistent, but very importantly, and if you bring your eyes back down to the graph on the left-hand side here, we have a very well characterized and understood signature for ERAP1 inhibitor target engagement. So when you inhibit ERAP1, you're preventing this peptide substrate cleavage, and a product of that is the fact that you get presentation of proportionally longer peptides bound to HLA at the cell surface. And this is exactly what we can see here. So those peptides that decrease in copy number when you expose the cells to the drug are generally those 9-mers, and those peptides that increase in copy number when you expose the cells to the drug are generally those longer peptides. So these 10, 11, 12, 13, and 14-mers. And again, nice to see that when you take that significant set of peptides from the Spectronaut 20 search, that we can recapitulate that biological signature that we know is a biomarker of target engagement for ERAP1. And really, this final slide just summarizes everything that I told you across the previous few slides, but in a slightly different visual. So on the left-hand side here is just a heat map displaying all of those individual peptides and the clustering of those peptides using hierarchical clustering across the top. And again, we can see in the Spectronaut 19 search that we get really nice dose-dependent separation of our different drug concentrations. And if you cast your eyes to the top right-hand side of this heat map, you can see that those peptides that are relatively lower abundance in DMSO that increase in abundance with increasing concentrations of the drug are generally all of those peptides that are 10 to 14-mers. And then the opposite is true for 7 to 9. So 7 to 9 start higher and decrease in abundance with increasing concentrations of the drug. And what is nice to see again is that if you then do exactly the same to those significant peptides in Spectronaut 20, although we can't change it, we can't control the directionality of the clustering here. And in fact, the clustering is not only switched at the top, but also switched at for the rows as well. We can still see a consistent biological signature we can see in Spectronaut 19. So again, these are the peptides here that are relatively lower in abundance in your DMSO samples. They proportionally increase in abundance with increasing concentrations of the drug. And those are those peptides that are 10 to 14-mers in length. So really, to summarize, based on the first case study, we've seen what looks to be quite significant improvements in library-free based directDIA searches, and really only modest improvements when you add in that supplemental 10-fraction DDA library in the context of this experiment. But it does seem to be that that increase in library-free peptides both have consistency with Spectronaut 19 peptides, but also enable us to recapitulate biology that we know to be there and seem to be biologically relevant for the cellular system we're studying. So this really gives us the first bit of confidence about starting to think about the potential of moving away from the requirement of generating a spectral library for these experiments. So to move on to the second case study now, And this case study is not a preclinical case study, but we also use immunopeptidomics to understand the level of target engagement we're getting with our inhibitors in the clinic, so very much through a translational lens. And we'll take you through the evaluation of Spectronaut 20 relative to a deep PBMC external spectral library we generated to search clinical samples with. So prior to starting any clinical assay, it's always good to ascertain the appropriateness of that assay relative to your clinical settings. So what the left-hand side here is describing is a ramping study that we performed in 3 independent healthy PBMC donors, where we took 2 A2 positive donors, 1 A2 negative donor, and we had aliquots of 5 million cells all the way up to 25 million cells. And the outcome of this experiment was to really try and understand what the level of PBMC input would be and what that PBMC input would give us in terms of peptide IDs at a 1% FDR. And that is what the graph on the right is showing you here. And I've included Spectronaut 18 in this because Spectronaut 18 was a software release at the time of conducting this experiment. So as you can see, as you increase the number of peptides that go into this assay, you also increase the number of as you increase the number of PBMCs that go into this assay, you also increase the number of peptides that you recover at a 1% FDR. But if you track this green line, this still gave us quite some motivation to try and improve both depth and coverage of this peptides, particularly when searching our clinical samples. And one way we thought about doing this, and largely because of insufficiency of clinical material to make a spectral library on a per patient basis, was to make an external reference library that we would search all of our clinical data against. So again, left-hand side here, we sourced ex vivo healthy PBMCs that were HLA matched to the geographies that we were running our trial in. And because they were ex vivo PBMCs, we were also able to stimulate these with different concentrations of drug in vitro. And then we were able to use these cells to generate a highly fractionated spectral library that we call deep PBMC. And the graph on the right here is just showing you the amount of peptides that were in that library for both Spectronaut 18 and Spectronaut 19 searches. And this deep PBMC approach turned out to work pretty well in practice. So at the time of making this slide, we'd acquired immunopeptidomics data across 131 samples in our clinical study. And across all of those 131 samples, we had a median increase of about 26% in peptide IDs. So this is when our DIA samples were searched against this deep PBMC library relative to a library-free search. And what you can see from the graph on the right-hand side is although we generally always improve on peptide IDs with deep PBMC, those results are more variable when we're in lower quantiles of PBMC yield. So quantile 1 here really describes the quantile that has the lowest input or PBMC input into this assay. And what you can see is there's a positive correlation with the amount of PBMCs we put into the assay relative to the amount of gain that we get in peptide IDs when searching with this deep PBMC library. So when we were invited to beta test Spectronaut 20, we thought this would be an excellent data set to revisit and do some retrospective analysis on. And this is what I'm showing you on the left-hand side here. So we went back to revisit this titration experiment, and quite again encouraging to see is that this purple line is the IDs for the Spectronaut 20 search. And even by the time we're at 10 million, the amount of peptide IDs that we're recovering at a 1% FDR was already exceeding that of 25 million input for Spectronaut 19. And again, to show consistency with what we observed preclinically, is that these peptide IDs are not just spuriously identified, they're also quantitatively complete. So looking at these two upset plots between Spectronaut 19 and Spectronaut 20, we can see that this first bar here is all of the 4,000 peptides in this sample that had quantitative values across each of the titrations of cell amounts. And then quite logically, the next largest set had everything in but 5 million, because 5 million obviously has the lowest number of peptide IDs. And then if you work your way down this, 10 million drops out and 15 million and so on. So this was really, really promising to see, and to sort of put some numbers to that just below it. Across all of the 3 donors we collected in this titration experiment, the percentage increase between Spectronaut 19 and Spectronaut 20 was anywhere between the region of 37% to 48%. So we again took our deep PBMC library back, and the graph I'm showing on the left-hand side here is illustrating that. We took our deep PBMC library and searched it in Spectronaut 20. And again, interestingly consistent with the preclinical observations, we only get a relatively modest performance in the peptides identified in that library. So around a 10% increase in peptide IDs relative to Spectronaut 19. But what was very interesting to see is the graph on the right-hand side. And if I draw your attention to the orange and purple bars here. These two bars are the Spectronaut 19 directDIA, so library-free searches, and the Spectronaut 19 deep PBMC searches for one of the clinical cohorts that we collected recently. And you can see in almost all cases, with the exception of one or two samples, we increase the number of IDs that we get with this deep PBMC library in Spectronaut 19. But what is very interesting to see is that when you retrospectively go back and search this data in Spectronaut 20, you can see even with the library freebase method, we're getting a significant performance increase on top of what we would have ascertained or what we would have obtained with our deep PBMC library method. And then what is also interesting, building on that, is that now when you add in the deep PBMC library into Spectronaut 20, the performance increase is negligible. So we've gone, again, to put numbers to this, we've gone from a scenario of using our deep PBMC library to gain a 17% performance gain in this particular cohort in terms of peptide IDs, through to a library freebase search, which improves by 59%, and then adding that deep PBMC library only improves by a further 1%. And that was really interesting for us because we wanted to start to think about what these sets of peptides look like if there was only a 1% difference between them. So the graph on the left-hand side here is really showing you those search numbers mapped to their relative inputs in terms of PBMC yield. And you can already see that we're across all searches, we're generally doing better than we expected to do relative to that PBMC titration study. But what is really interesting is on the right-hand side is you can take this set of peptides that are identified with the Spectronaut 20 directDIA library free search, and you can overlap them with the set of peptides that are identified in the Spectronaut 20 deep PBMC search. And what was interesting for us to see is that these are really not diverging sets of peptides. So the library freebase method in Spectronaut 20 is actually able to recover a really significant proportion of all of those peptides that were being found with the deep PBMC library. So really, to conclude on this second case study, it seems to be that Spectronaut 20 now has, particularly in library-free mode, has searches for our translational data that parallel those of what we can find with this internal highly fractionated deep PBMC spectral library. Finally, then, to move on to the third and final case study, and this was really based off what we'd observed in the previous two case studies, and really building out evidence that Spectronaut 20 may be sufficiently good in library free-based searching that we may not need to make spectral libraries in some experiments. And two experiments where making a spectral library in particular was very cost and time prohibitive for us were some recent PDX models that we generated. So these are patient-derived xenografts implanted in mice. And for these particular experiments, and I'll show two of them, it took us about 3 months to grow sufficient cells just to make the spectral library alone. And that equated to about 50% of the time of the total experiment. So really to sort of bring your attention to the graph on the right-hand side, again, if we start with the orange and purple bars, you can see the Spectronaut 19 searches in library free. And you can see there's an additive benefit of having that spectral library in there in terms of peptide IDs with Spectronaut 19. But what is interesting to see is that when you retrospectively search this data with Spectronaut 20, you can see that you have a significant increase above and beyond what we were able to achieve with the Spectronaut 19 hybrid search. And again, then there's a marginal increase with the Spectronaut 20 when it's given the hybrid library as well. And just to show that again across another PDX study that we conducted, again, a very similar time to grow out these cells, approximately 50% of the experimental time just growing the spectral library. And again, you can see in this, if you focus on the orange bar here, is that the direct in this particular experiment, there was a significant benefit of adding in that spectral library, both in Spectronaut 19 and Spectronaut 20. So we identified almost twice as many IDs with the library as we do in the purple bar as with the orange bar. And interestingly, not as great a performance in Spectronaut 20 library free searching here. So actually the number of peptide IDs goes down in the library free method of 20 relative to the hybrid search in Spectronaut 19. And again, there is a further supplemental benefit in the Spectronaut 20 hybrid library. So slightly variable results across these two experiments, but really the probably beginnings of evidence for us to start to think about when generating a spectral library is very time and cost prohibitive, how we may think about leaving the spectral library out of the experimental design. With that, I'd like to wrap up and just really give thanks to everyone that's in this photo who I get to work with on a daily basis. And also thanks to Biognosys for inviting us to do some beta testing on Spectronaut 20 and having us give this talk. And please do reach out if you have any questions.